Hello everyone, and welcome to this first video podcast for our online lab this semester. For every podcast, I'm going to begin with a brief outline for that particular podcast. Today I'm going to start by going to the Blackboard website and just going over where everything's at so you'll know where to find things. Then I'm going to go over our teams. I'm going to go over some basic syllabus lab requirement stuff, and then we're going to overview the different lab modules briefly, and then we'll talk about Lab Module 1, which will take about 7 labs to complete. Then I'll close with what we're going to do for today's lab. As I'm sure you've noticed, you have 2 separate Blackboard shows for this course. One is for the lecture component, and the other is for the lab component. So what we are looking at here is the lab Blackboard show. And you're pretty familiar with Blackboard shows by this point. But just real quick, we have our syllabus here, which you can download. This is specific to the lab. So over here we have our lab manual. This has all of the labs we're going to do this semester in it. It's designed for the face-to-face labs. So there's going to be some deviations to it, but we'll still use it as we go forward with this online lab. However, as you see here, I point out, don't use the worksheets that are found in the lab manual. Rather, use the worksheets that are found under weekly materials. Last tab I want to go to is this weekly material. So each week there will be a separate folder. So for week 1, which is this week, when we click on that, we'll see a link for the quiz. So you click on this and it'll be a single quiz that's linked to labs 1, 2, and 3. And we'll talk about quizzes in just a moment. So we pull up lab 1, for instance, we'll see a PowerPoint slide. And that's the same PowerPoint that we'll see just a moment as I make this video. In addition, you'll also see a link down here for the video podcast that goes over that PowerPoint slide. That is the one we're making right now, so I don't have the link there yet because we're still making it. Also in the week 1 folder, as all the weeks will have, you'll see separate folders for each of the labs for that particular week. You'll also see this folder here called Week 1 Worksheets and Submission Links. So when you click on this, you'll see a Word document here that has all the worksheets that you'll be doing for that particular week. You can go ahead and click on it. And what you'll see is the several worksheets linked to each of the labs for this week, all in one place. What you'll do is you'll answer each of these questions on the Word document itself and save it. And then you'll go back to this Week 1 Worksheets and Submission Links folder, and you'll click on Week 1 Worksheet Submissions. And you'll upload this like you've done probably for other classes. You'll go browse my computer, and then you'll just upload that particular document. Every member of your team needs to do their own worksheets. I expect you to work with each other, but you have to have your own answers. And that's all I wanted to talk about in regards to the Blackboard shell. Now I'm going to go back to the PowerPoint slide. So let's continue our discussion, and let's talk about your teams. These are the teams you'll be in for lab. You'll notice they are the same teams that you are in for the lecture. I don't always make the lab teams and the lecture teams the same teams. However, with this being an online course, I thought it would be more helpful to have the same members of your teams. That way, if you needed to coordinate times to meet, you didn't have to worry about your lecture team versus your lab teams. So I think this will work out a little bit better. You will meet for your labs using the Teams application from Microsoft that we use for lecture. When you meet with your teams for the labs for this semester, you can choose to meet at your assigned time that was posted in the registration. Now, the advantage to meeting during your regular scheduled time is that you will have access to the TA or myself. One of us will be there during your lab time. However, I did want to provide a little bit of flexibility for the labs just in case there was a member or two that had some other obligations that they would prefer to meet at a different time. The only catch to that is that you have to make sure everybody in your team is available at that alternative time. We can't leave anyone out. Now, when you do meet, either during the regular time or a different time, you'll use the same Microsoft Teams application that we use for lecture. You'll go in the exact same team channel. Some advantages to completing these labs using Teams is that you can save documents right there in Teams and you'll be able to access them later. You can work on those simultaneously, and of course you can share your screen with each other, so if you're working on a particular lab activity, you can do that together. Okay, let's go on. There are a few things required for this class. A lot of it will be found on Blackboard, as we already talked about, but you will need the Writing Papers in Biological Sciences. The 5th or 6th edition is fine. It's the same one that you had for Biology 111 and Biology 112. So hopefully you saved it. The lab manual is found on Blackboard, as I've already shown you. As I just want to remind you again, it is written for the face-to-face lab, but we're going to use it as our same general theme, but there will be some adjustments that we have to make, and I'll let you know when that happens. And again, just to remind you, don't use the worksheets found in the complete lab manual, rather use the ones that are found in the weekly material. Completed worksheets, as I said before, will be submitted on Blackboard using the assignment link. As I showed you on the lab Blackboard pages, there are links for the podcast and the PowerPoint slides under the weekly material. Okay, let's talk about grading for just a little bit here. I want to remind you that in order to move on into biology curriculum, you have to get at least a C- in the entire course, lecture and lab combined. And an additional stipulation is that you can't fail either the lecture or lab. That means you have to at least get a 60% in both of those. Now together it has to be at least a C-. And considering that, remember that the lab here is worth 30% of your grade. Any assignment you get, this is true in lecture as well, but in lab too, you have 72 hours to dispute your grade. Make sure you just email the TA if you have something you want to dispute in lab. Late lab assignments will have a 25% deduction per day late, and that includes the weekends as well, as it says here. The first 25% comes off the minute it was late, and then the next day it would be another 25%. Now, if you have an assignment that's going to be late and there's an acceptable reason, make sure you reach out to your TA and myself ahead of time, if you can, to let us know. Because I understand that certain circumstances come up and, well, things just happen. We get that. I've already mentioned this a little bit, but let's just say it again. You must complete labs with your lab partners. So it's really important that you communicate with each other to find a time that works for each of you. Now, there are a few things, of course, that you have to work on yourself. These include the quizzes, which you'll have weekly. You'll have an introduction to write, and so that final copy of that introduction needs to be written by you. You'll see that as we move forward that there will be an opportunity to collaborate with each other as you do some peer editing, but that final introduction needs to be in your words. Same is true with the final complete research report. Now let's talk about lab quizzes. These will be administered through Blackboard, and I already showed you the link for those. You'll have one quiz per week. During the regular semester, we would have a quiz that is due at the beginning of each lab. Here I'm making one quiz for the entire week of labs, and it is due that Thursday at 11:59 p.m. However, I really think you should do it before you actually do the labs themselves. Okay, let's talk about the lab modular setup. The lab is broken up into 5 modules. In each of these modules, we'll work with a different model organism, except the 5th one, which is not really a model organism. The first one is plant biotechnology. Over here is Arabidopsis. It is a model organism used for plant research. As you can see here, that this particular lab module will take up a lot of time. It is 7 specific labs, and the end product of that will be an introduction. Now for the second lab module, we'll focus on C. elegans. It's a microscopic worm shown here, and it is a fabulous model organism, and we're going to use it in this lab to look at data on behavioral genetics. There are 5 labs devoted to this, and the end product here will be a complete lab report. Our third lab module will be on Saccharomyces cerevisiae, the budding yeast or the brewer's yeast. In this one, we'll look at data involving a genetic screen to identify genes necessary for stress resistance. And you'll develop a poster for this lab module. And then Drosophila fruit fly genetic cross lab. This one is only one lab, and in this one we do simulations. And in fact, we do these simulations even in the face-to-face lab environment. So this one translates very well to the online lab. And then the 5th module is a debate. Now I'm going to do something a little different for the online lab, and I'll talk more about that later in the semester. All right, now let's talk a little bit about Module 1. Remember, this is one that's going to take 7 labs, so we spend quite a bit of time on this particular module. Each of the modules are broken up into specific lab days. So we have 1.1, 1.2, 1.3, and all the way down here to 1.5. You'll see in this module that we have a day devoted to working on writing the introduction and also developing the first figure legend. And then we have a day here devoted to peer editing the introduction. And so these aren't specific lab days, but there are days for writing. You'll still work with your groups, but it'll be more so that you have time to really fine-tune that introduction. Today, you'll see that it's just an introduction to the virtual lab. We'll introduce Module 1, which I'm doing right now, And then there's this pipetting skills and simulation that's underneath this red bar here with a quiz at the end of it. And I'll talk about that in just a moment. Module 1 class goal here is to isolate this gene, the GAPC gene, and then sequence it. And then what we're going to want to do is compare it to other known sequences. Now the role of GAPC, it's a glycolytic enzyme that you probably learned about in cell biology, probably last semester. It catalyzes the 6th step of glycolysis. I'm sure that is your favorite step of glycolysis, because why would it not be? Now, as I said, we're going to sequence kale here, but we already know the sequence of wild cabbage and Arabidopsis. And so we're going to use these two known sequences to compare it to the new sequence. Now, the enzymes of the glycolytic pathway are very well conserved, meaning that the differences in their gene sequences have not changed much throughout evolution. So we say it's very well evolutionarily conserved. And so we can use different species within plants to compare it. In fact, we could compare it to ours, but I suspect it wouldn't be much different than the sequence found in our own genes. Now, what we can determine by looking at this figure here is the evolutionary relationship between members in the Brassica and the Arabidopsis families. We can see that kale and cabbage, they're both found in the same Brassica group here. We can see that Arabidopsis is up here. So it's in a different group. We might be able to make some kind of prediction before we even start this about how similar kale is going to be to cabbage and how similar it's going to be to Arabidopsis. We expect there should be considerable conservation between all of these because they share a common node right here. This is where they last shared a common ancestor. So starting with lab 1.2, which will be our next lab, you'll learn about how we extract DNA and we'll learn about PCR. Then the next lab, we'll talk about how we analyze the PCR results using gel electrophoresis. And then we'll talk about the importance of doing this second round of PCR that we call nested PCR. We'll analyze the nested PCR results in this lab 1.4. And then we'll talk about how we sequence the DNA and how we will compare the DNA. All right, for today's lab, there's just a couple things we're going to do. The first is that you're going to make an account with Lab Exchange, and here's the link shown here. It's a free account, and it's offered through Harvard Education. It provides a lot of really nice simulations and videos, and we'll take advantage of that for a few of our labs. Not all of them, but a few of them. It's also free, so that makes it even better. If you haven't done so already, all you do is go to their website here, and you click on Sign Up, and you just go through the standard sign up procedures. And once you've done that, I get a message asking me to approve your account. And so I approve your account, and then you have access to the material that I've provided through Lab Exchange. Once you have been approved, then what I'll need you to do is go over here to Browse. You click on Browse, and then you click on Dashboard. And once you've clicked on Dashboard, you can see a few things here, but where I want you to go is Classes. And under Classes, click on Biology 211 Genetics Lab. And you'll see a few things I've already placed here. And the only one that you're going to use today is this micropipetting solutions. And so what you'll do is you'll click on that. You'll hit Start Simulation, and then it'll take you through the simulation. It's pretty good, I think. It's pretty detailed. Nothing's as good as doing the real thing, but this provides some of the key steps of how to use a pipette and where students go wrong with using a pipette. As you're going through the simulation, it doesn't really let you get away with doing shortcuts. Like, you have to open up the lid before you take the liquid out. If you try to use the same tip going into different solutions, it's going to say you can't do that, and it's going to make you throw it away. When you go up here, maybe you can't tell, but these are pipette tip boxes. You got to open up the lid first before you can get them out. Sometimes it's a little annoying, perhaps, but it's pretty real, though, right? Because these are things you have to think about when you're working in a lab. Closing and shutting tip boxes seems incredibly boring, but also incredibly important. So you'll go through this simulation, and then at the end of it, there's a quiz. The quiz is pretty straightforward, and the good thing is it'll let you keep answering until you get it right. However, try to get it right on the first time because it's a little bit more fun that way. Another thing I want to tell you, I forgot, I should have mentioned just a second ago, but I'll tell you now. When you're working on the simulation, you'll see in the upper right corner a lab notebook, and it will walk you through step by step. Make sure you follow each of those steps. If you try to bypass it and jump ahead, it'll make you go back, or it'll make you start all over again from the very beginning. So make sure you go through steps. Another thing I ran into when I was doing these is sometimes it's asking you to use a particular piece of equipment. With the gel electrophoresis, for instance, it was telling me to use the centrifuge, but I couldn't find the centrifuge, and that was because it was behind the lab notebook. So sometimes you have to shut the lab notebook in order to reveal what's behind there. Well, that's all I'm going to say about Lab Exchange, but sign in for it, I'll approve it, and then you can start your simulation. Okay, so here I write out exactly what you need to do. I'm not going to read this to you because you can read it, but the PowerPoint slides are available for you. So as you're going through this, if you're having problems getting logged into it, just go through these steps and all should work out well. But if it doesn't, please let me know. Okay? I mentioned the simulation that you're going to go through. The simulation, make sure, I forgot to say this before, you sign in as level 1 for the simulation, and then you'll have these 6 different categories to go through. Once you've completed this, you get the 13 points that are allotted for this assignment. Each member of your team needs to do this. I would recommend as you're in the teams, recommend as you're in the team's meeting space that one of you shares the simulation on the screen and the other work through it at the same time. That way you're doing it together. Okay, looking ahead to the next lab, make sure you read these following sections, Labs 1.2, Part 1 and 2. Prepare for the lab quiz. Now, the lab quiz, as I said, is all one. That's open at the beginning of the week. But one thing you might do is think about that each of the labs, most of the labs have 5 questions for them. You may answer the first 5 questions, leave it open, don't submit it yet, leave it open, and then go back to it and answer the next 5 questions, and then the next 5 questions. Alternatively, you can just go straight through the whole thing and just answer all 15 questions at the same time. It's really up to you, as long as it's done by Thursday at 11:59 p.m. Okay, that's all I have for this first video podcast for our online labs. If you have any questions at all, please make sure you contact myself or your lab TA. All right, I'll see you later. Bye.